I am so excited about this part of the lecture. In fact, this is one of my favorites. I probably said that before, but this is one of my favorites to do because I think it is just so incredible that as humans, we have been able to harness this biological power of the body to create a medical advancement that has saved more lives than any medical advancement to date. And that is vaccination. So vaccination, we're looking at ways that we can prevent diseases. It is a routine medical procedure. Often people don't think of it as a medical procedure, but it can be classified as that. And it provides people with memory or the immunological memory of a pathogen without ever having to be exposed to that pathogen, or at least infected and having to fight it off on your own. So you gain the memory, all the benefits of memory, without having to suffer any of the consequences of actually getting the infection. This is achieved by immunizing people with a non-infectious vaccine. It contains the antigens that stimulate the immune system, but depending on the type of vaccination, those antigens are going to be either in pieces, or they're going to be part of an activated pathogen, or they're going to be created by the components of the vaccine. Regardless, anyway, those antigens are able to stimulate the immune system, but you never have to have the pathogen actually infect you. Typically, 2 or more doses are given, and that's to mimic what would happen in the actual immune response. So the first time a dose is given, it is like the primary response, which takes time, builds some antibody, but wears off for a while. Then a second dose or a booster is given to mimic that secondary immune response, which is much more powerful, stronger, and you get better, stronger immune responses than with each subsequent infection. And so therefore, after immunization, if you're infected with that pathogen post-immunization, that pathogen is going to be eliminated so quickly that it's not going to cause actual disease. And like I said, it is claimed that vaccination has saved more human lives than anything we've had so far in medical breakthroughs. Oh, I was hoping for a good smooth transition there, but I guess we need to click on the slide. There we go. What's the history? Why call it a vaccine? It seems like so out there, right? Well, it actually began many, many, many years ago in the 1700s, late 1700s, Edward Jenner. So there was smallpox going on, and Edward Jenner noticed that, well, I'm sure a lot of people noticed it, but that cow maids, so the women who milked the cows, would get a less severe form of smallpox called cowpox, and it didn't kill them, but they were never infected with smallpox. And so Edward Jenner then published this report on how material from animals infected with cowpox could be used to protect against smallpox. And this innovation then used cowpox virus. The name of cowpox virus is vaccinia virus, where now we know where vaccine came from the name of the cowpox virus. It causes just a mild infection in humans, but it looks very much antigenically like the smallpox virus. And the immune system would fight off cowpox, not really have much of a problem, but develop a really strong memory response. And then if those individuals were ever exposed to smallpox virus, it looked enough like cowpox that the immune system memory kicked in and would wipe it out. Now, that was in, like early 1800s when this was all happening, and a lot has happened since then. Smallpox virus or vaccine was the first to be given, and it's also the only disease that has been completely eradicated from human population. It was declared eradicated in 1979. So we're looking at, like 200 years later. So it takes a long time for a virus to actually, or a pathogen to actually be eradicated. But smallpox vaccine is not given to children any longer. So I'm not sure what year they stopped giving the vaccine, if it was in 79 or if it was in 80, but it is no longer routinely given to children. Now, we did have biological warfare. Smallpox was known as a pretty potent biological warfare agent. And so some people have been, well, we have a huge population still alive that's been vaccinated against smallpox, but anybody born after the late 70s probably is not immune. And many of those people were in the military age group. And so a lot of them, since biological warfare has become, a hot topic, some people, especially in the military groups, are being vaccinated against smallpox again, but I don't know. It's definitely not done for the regular public. In fact, here I have a breakdown of what is done for recommended by the CDC. This was pulled from the CDC website now. So this is current as of 2023. And it has a list from birth to, this one goes through 4 to 6 years, and the different vaccinations that are recommended for babies or for children. And you can see that right away, hepatitis B is recommended right out of the bat. And then we have a whole bunch of them that happen early on. Rotavirus, DTaP, which is a mixture of tetanus, diphtheria, and pertussis, whooping cough. Haemophilus influenza B protein, pneumococcal. Those are both Haemophilus and pneumococcal are going to be like a meningitis or a pneumonia vaccination. And then polio is still given to children very early in life. And then you can see that there's boosters throughout the first year of life, and then sometimes up into early adolescence. Now, past 5 or 6 years, there are other ones that are recommended. Every year, adults and children are recommended to get the flu and COVID. Now, that's a brand new one out, COVID-19 vaccines. And the MMR is started at about a year, as well as the chickenpox virus, varicella. Hepatitis A can be given as well. It's not required, or I don't believe so, but it is recommended. And then before middle school, right? Oh no, this is still 4 to 6 years, sorry. MMR, varicella boosters are given. Now, if we go a little bit older, into later adolescence and into adult years, MMR is going to be given a little bit later, right before maybe middle school area, as well as HPV is another one that is given to those kind of in the middle school age. Meningococcal, usually that's a meningitis as well, usually given before attending college or going into the military. And then depending on if you still need a hepatitis B or hepatitis A, you can get those throughout adulthood. And then there's going to be some as you get older, beyond 18 years, that are recommended for later adults, like the shingles vaccine. We'll see less and less of that, though, because people now are getting vaccinated against varicella. So people aren't getting chickenpox early in life. And so that's a limited time vaccine for shingles. And there are some other meningitis and pneumonia vaccines that are given to older adults as well. So vaccines really are big early on in life. and then they get less and less as you get older. But that's because you're exposed to pathogens at a young age, and you want to make sure you don't get actual infections of those before you get vaccinated. Now, there's lots of different types. So these are the different diseases that can be prevented by vaccination, but there's lots of different types of vaccines. And so we're going to look at each one of these. Some of them we kind of group together because they're kind of put together anyways, but we do have a variety of different types. And with those vaccines, the different diseases, they're going to kind of fall into these vaccination types based on the type of antigen that we're looking at or the type of pathogen that we're looking at. So let's go ahead and walk through each one of these and talk about them just a little bit more, a little bit about each one of them. So you also have to remember that we're looking at viral vaccines as an intracellular pathogen, and then bacterial pathogens as being external to the cell. And remember that they are processed differently and presented differently, MHC class 1, MHC class 2, if they're internal or external. Okay, so that's also going to determine what type of vaccine is utilized. With most viral vaccines, it is important that the virus is in its native form, because that's the way it's going to be most antigenic. But we don't want the virus to be in an infective stage, or at least a normal infective stage, because we don't want to end up giving somebody the actual disease by giving them a vaccination. So to do this, there's really kind of two ways that we can approach viral vaccines, keeping the virus intact. One of them is by inactivating the virus or killing the virus. I don't really like killing the virus, and that's just because viruses were never alive to begin with. So inactivated virus vaccines tends to be the more approved or more utilized term. But it consists of the actual viral particle, but it's been inactivated in a way, whether it's been heat or by some sort of chemical, that it's not able to reproduce. Even if it'll get inside of the cell, but it won't be able to, replicate once it gets inside of the cell. But it can still generate an antibody response. And so polio, the injected polio virus, influenza, rabies, these are all examples of whole virus particles that are given, but they're inactivated. They're not able to replicate. Now that's good. Now polio and rabies are two viruses that we definitely don't want to have any chance of them replicating, because they can be very, very detrimental to the health of the individual. Influenza as well kills a lot of people, probably not as detrimental as rabies and polio to the larger population, but also one we don't really want to have able to replicate. So it's good to have them killed. It works. Those types of viruses, even in their killed or even in their inactivated stage, still are able to mount an immune response. The body's able to mount an immune response against them. But a second type of viral vaccine that is probably a little bit more effective is the live attenuated virus. What this means is the virus is actually in its full native form, and it still is live, and that means it's able to replicate, but it's attenuated in that it doesn't cause disease. So it's not pathogenic to humans. Very effective because it looks so much like the actual pathogen that the immune response is very strong against it. Examples of this would be like the oral polio virus, the one that you drink. Measles, mumps, those are both viruses that are live yet attenuated. Let's look at how an attenuated virus is created. They do provide or elicit a more robust immune response. because they really truly mimic what a real true infection would look like. And so the immune response can be very appropriate against it. But in order to make sure that it's not going to be infective, the virus is attenuated, usually by growing it on human cells, and then using it, infecting other related type cells from a different species. And therefore, there's going to be an incorporation of DNA that allows mutations to occur in the DNA, or in the DNA or nucleic acid. It could be an RNA as well, virus. But those mutations make the virus unable to really infect human cells anymore. So here, this picture is showing where it's using monkey cells. I know pig cells have been used. There could be other, and it's not inside of the animal, it's just on a cell culture. So we're not using an actual animal to make these. But after being grown with another type of species, is no longer infective, or at least no longer going to cause a disease in humans. So anyways, that is one way that viruses can be attenuated. Very strong response that the immune system can generate against a live attenuated virus. And so one of the best forms of a vaccination. Now, subunit viruses, now we're still, or subunit vaccines, we're still looking at viral pathogens, can utilize little fragments of the virus to actually stimulate an immune response. Not as strong as using a full virus, and not as strong as using a full attenuated virus, but yet still effective. Hepatitis B is an example of this type of vaccination. The subunits are actually created using recombinant DNA technologies, where the DNA to create these proteins is made in a lab, and then the proteins are produced from that DNA. And so the hepatitis B subunit vaccine was the first of this type of technology. And the cell that's used to make these proteins is a yeast cell. So a yeast cell is a eukaryotic cell, so it'll have a lot of the same genetic machinery that the human cells do. And those little DNA coding regions are put into a section of DNA, and then the yeast grows up this protein in mass amounts. And then it can be injected into or put into a vaccine. So that's a pretty great way to use a protein rather than just a full virus. Another type of viral vaccine that definitely has made a lot of attention in the public lately has been nucleic acid vaccines. Nucleic acids, either DNA or RNA. Both of these technologies exist. The only nucleic acid vaccine that we have out approved by the FDA is for SARS-CoV-2. It was a big deal to get this approved in the time it did, but know that nucleic acid vaccines have been under research and under development for over 20 years. So it's not like they just pulled this out of, oh, let's just do this, and had it done in a year and a half. That's not true at all. What's pretty great about the nucleic acid vaccines is that rather than delivering the full vaccine or the full virus or a subunit, just the instructions to make the antigen is delivered, and the host cell makes the protein that is antigenic, and then the immune response will come about because of the antigens that are produced by the host. And so the code is actually inserted into human cells, and then the human makes the proteins. The immune system recognizes the proteins as non-self and mounts an immune response. And then when the actual virus when the body's exposed to the actual virus, there's memory for those proteins. So nucleic acid viruses, although have been in development for a long time for a variety of different types of vaccinations, it was a great time to utilize this technology to rapidly produce and mass produce a vaccine that we needed to curb the pandemic. And we can talk about this more in class if you want to dive more into it. But it's a pretty novel way or approach to making a vaccine. So this approach then offers a number of potential advantages over the traditional give the actual pathogen type of approach, right? So there's both B and T cell responses that are stimulated. Nucleic acid vaccines can have a pretty good stability. Some of them can even be at room temperature for a while. There's absolutely no risk of getting the infection because the virus, the actual infectious agent, is not being given. And it's relatively easy to manufacture at mass capacity and scale at a very large manufacturing scale rate. Really, though, nucleic acid vaccinations are only going to be useful for certain pathogens that have that type. It's going to be a virus, and it's, the nature of the infection itself is going to dictate what type of vaccine is going to be best, and then what type of immune response is required. So SARS-CoV-2 was a really great candidate for this type of vaccination. Now, other types of vaccinations were developed for SARS-CoV-2, but the nucleic acid was the one that got first FDA approval. Okay, so that was kind of about viral pathogens. Now let's switch and let's talk about bacterial pathogens. So the development of vaccines against bacterial pathogens are similar in that antigens have to be delivered in some form, just like with viral diseases. But those antigens can be part of the bacteria itself, but it also could be a toxin that the bacteria produces that is the actual antigen. And so some bacterial diseases are caused not by the bacteria itself, but the actual the proteins or the toxins that are produced by that pathogen. So diphtheria. Diphtheria is a bacterium. It's called Corynum bacterium diphtheriae. And it is not infective, but rather it produces diphtheria toxin, which is the cause of the symptoms that we see in the respiratory tract. Tetanus, another one, caused by the bacteria Clostridium tetani. Clostridium tetani produces tetanus toxin. Tetanus toxin paralyzes muscles. The bacteria itself doesn't do anything. For toxins, the best way to prevent them from causing pathogenesis is to neutralize them. And so having a very strong neutralizing antibody is the way to approach protection against these diseases. So lots of times vaccines for bacterial toxins are going to be made by purifying the toxin and then treating it in some way, maybe chemical, to remove the toxicity. So it still looks like the toxin to the immune system, but it doesn't have the toxic activity that it would if it was not treated. And so we call these proteins inactivated toxoids. So the toxin would be the activated or the pathogen causing or the disease causing form, where a toxoid is inactivated and it is not able to actually cause disease, but it can still elicit an immune response. Now, some bacteria produce a capsule on their outer covering. And this capsule is made up of carbohydrates, polysaccharides that are going to be species specific and really are the antigen that is recognized by the immune system. The capsule then can also be pathogenic and antigenic. So this is a case where the bacteria itself is pathogenic. It's not the toxin. Pneumococcus, meningococcus. Those are types of bacteria that have capsules. And then Haemophilus influenza, or H flu. Streptococcus pneumoniae is the first one. Anyway, they have this capsule that needs to be, that is the antigenic portion, and needs to be recognized by the immune system. But the capsule prevents complement from being activated. And antibodies, so, and this is through the alternative pathway because we're looking at the innate immune system. And so only when antibody have bound to the capsule will complement be allowed to be activated, and then the bacteria can be cleared. So then the aim here is to produce high affinity antibody that can bind to the capsule and activate complement. It's difficult with capsular pathogens, definitely, but protection against bacteria with a capsule is provided by high affinity IgG that have the ability to bind to polysaccharides. Remember, B cells can recognize polysaccharides, and so they can make antibody against polysaccharides and then trigger this complement activation. Making this high affinity IgG antibody requires that B cells are engaged, sure, but remember that B cells can't do anything without the help of T cells. But T cells can't recognize polysaccharides. Oh my gosh, we have an issue here, right? How are we going to get a T cell to be stimulated by a carbohydrate? We're not going to, because that doesn't happen. So a vaccine against a carbohydrate, like we see on these capsular bacteria, needs to be combined with some sort of protein epitope or some sort of protein antigen. And so this led to the development of what we call conjugate vaccines. Conjugate vaccines have different epitopes that are conjugated together, that B cells recognize the carbohydrate portion, T cells recognize the protein portion. And so they're synthetically linked together, and often you're going to have a polysaccharide with some sort of inactivated toxin or a toxoid on the attached to it. So here's an image. It's sideways as compared to what is in your text. I don't have enough room on the slide. But the B cell has the B cell will have the IgM receptor on its surface that will recognize the polysaccharide portion, will internalize it, will process it. But since that protein toxoid is conjugated to that polysaccharide, that is also internalized, also processed, and then presented. B cell is stimulated by the polysaccharide. T cell is stimulated by the processed toxoid that gets presented on MHC class 2. Then the T follicular helper cell can drive the B cell into producing antibodies against the toxoid as well as against the polysaccharide. So this conjugate vaccine is important to get antibodies against carbohydrates, but also activating T cells. And so you actually get two vaccines in the case of one. So Haemophilus influenza, pneumococcal, meningococcal, those bacteria are all produced through a conjugate vaccine. So on immunization, dendritic cells then are able to process the vaccine present toxoid-derived peptides to naive CD4 positive T cells. And then those B cells will be able to interact with the follicular T cells that are driven by the dendritic cell activation. The naive B cells that have epitopes for polysaccharide, but also are presenting the toxoid, then can interact with the T follicular helper cells. So then there is the development of memory for the polysaccharides, as well as the toxoids, to make antibody that can be used to bind to these bacterial surfaces, these capsules, and drive complement activation. Okay, that's conjugate. Now, there's a term here when we talk about vaccinations or vaccines called adjuvants. Adjuvants are really things to get inflammation going, because we know that in a true immune response, there's going to be inflammation. Mucosal immunity is different. We're looking at systemic immunity here, non-mucosal surfaces. And inflammation is a really big player. But if only proteins are given as a way to stimulate the immune response, that's not going to be very inflammatory to the immune system. And so vaccines that contain these microorganisms, these viruses that are either inactivated or attenuated, they're pretty good at stimulating the immune system, but it's when the subunits, because they have everything, right? They're going to drive inflammation. They're a full intact virus. But the subunits and the conjugates, they only have pieces. And some of these pieces are very purified proteins, and they're not going to activate the innate immune response in any way, and you won't get that inflammation. And so then there's not going to be a typical type of response. And so for these subunit and conjugate vaccines, adjuvants have to be added to stimulate that innate immune response. Adjuvants are really nothing. They're just kind of inert substances, but yet they are stimulatory to the innate immune response. Now, the DTaP, diphtheria, tetanus, and acellular pertussis is what DTaP stands for. It's what we call a combination vaccine, because it's made up of all of these things with an adjuvant. There is diphtheria toxoid, so a purified inactivated form of diphtheria toxin, tetanus toxoid, inactivated form of tetanus toxin, and then acellular Bordetella pertussis. So it's pieces of the Bordetella cell, but it's not the full cell itself. And because of all of these things, you have the proteins and you have that acellular pertussis. That acellular pertussis actually acts as this adjuvant to stimulate the innate immune system, but also generate protective immunity against all three of these organisms or these pathogens. But some of these vaccines don't have that acellular pertussis to act as an adjuvant, so you have to add adjuvants to it. So what's this? Diseases for which current vaccines are effective for acute infections. I think this slide is out of place. Let's come back to this one. I want to jump to this one. Adjuvants. So aluminum salts, or alum, is actually the one major one that is utilized in the US. You can see that it is included in a whole bunch of different vaccines. Shingrix is a vaccine for shingles. That one uses an MPL, which is a lipid A protein. Other than that, there's really no other adjuvant outside of aluminum that's used, or some sort of aluminum salt. Again, it's not going to be activating anything else in the body except for the innate immune system, and that innate immune system is needed to get the actual memory going, to get the adaptive immune system going. You need that inflammation. Okay, vaccination programs is where I want to go, and I think that's what this slide was about. The diseases for which current vaccines, oh, you can't see, I gotta go back one more, nope, too far. The diseases for which current vaccines are effective are for really acute vaccines, or diseases. So like measles, influenza, any of the meningitis diseases. Those are all very acute, and we're able to fight those off really easily with vaccination. Well, not really easily, but we've been able to figure out vaccinations for them. Now, the diseases that tend to be more chronic in nature or last a little bit longer, but are very prevalent in the human population, we have a little bit more difficulty creating vaccines against them, but we're trying. They're out there. They're in studies. You know, we definitely have some HIV vaccinations out there. We have work with malarial vaccines, tuberculosis. These are big, huge diseases in the population, but we have a tough time creating a vaccination. We're getting there, but chronic diseases are a little more difficult than, say, those acute diseases are. And all of this, there we go, is happening through vaccination programs. So after an extensive vaccination program, the vast majority of the population will typically have protective immunity against a pathogen. Those individuals that get vaccinated more than likely will have protection. And if you have the majority of a population get vaccinated, you're going to have a huge mass number of people that cannot be infected, or at least not develop disease from a pathogen. And so this situation develops a population that has what we call herd immunity. This term has been all over the news, has really been highlighted since the development of COVID-19. And herd immunity is when a huge population has protective immunity that can protect those few people within the herd that don't have immunity, because there's not going to be, the virus isn't able to jump from person to person to person because it gets blocked. And so that protects those individuals who either cannot create an immune response, or have not gotten vaccinated, or whatever reason that they are susceptible. And so herd immunity breaks the infective chain. Now, if the proportion of children being immunized decreases, that is going to decrease the number of people in a population that have immunity, or rather increase the number of people in a population that are susceptible. And herd immunity can be lost. And that's pretty detrimental for those individuals who, for whatever reason, are not able to make an immune response. Maybe they have an immunodeficiency. Maybe they have some other reason that they're not able to mount an immune response, and they rely on herd immunity for protection. But if you have too many people relying on herd immunity and not getting vaccinated, you'll lose the herd immunity, and you end up with a susceptible population. And so vaccinations have really, really been in the hot topic of consideration in the news, in the media, in just people talking, because of COVID-19 and the way that vaccination was approached in that light, as well as all of the public policy, or the political underpinnings that seem to be aligned with vaccination. There's two big, huge differences when we look at the development of a successful vaccine as compared to a drug. right? People weren't going to take vaccination because there were drugs out there. Well, that's great. Drugs are given to people that are ill. And so it's easier for people to say, yeah, if I get sick, I'll take a drug. And any alleviation that somebody gets from that drug is considered to be good. Vaccination, on the other hand, is given to healthy individuals who never know if they're going to be ever infected. And oftentimes these healthy individuals are babies. And any adverse effect that comes about because of a vaccination then is to, a greater concern. And it's a greater concern to their parents than the benefits that the child might receive from getting the vaccination. So that's tough. It's tough where, drugs, you can see the effect. Vaccines, you can't always see the effect. Drugs are also prescribed on a case-by-case basis, and they're only given to somebody by a physician specifically for that patient for a specific disease. Vaccination is given to the masses. It's part of a large program that's often going to be mandated or at least recommended by some sort of authority. And we see issues with political issues surrounding authority, and this is becoming more and more prevalent. And so applying vaccinations to a population at large dictated by an authority tends to be an issue, where you don't see that with giving drugs for a specific patient for a specific disease. So vaccination programs are difficult to A, administer, and B, to gain the trust of the population. And so the only situation in which a vaccine benefit can be truly felt is when exposure is obvious. So you don't know how many times in your life you've been exposed to measles. Maybe 0, maybe 6. But if you've been vaccinated, you don't know because your body fought it off. And so unvaccinated members are the ones then that can succumb to suffering and disease, right? Or dying, right? Succumbing to the disease. So it's tough to say what the benefit is unless you know you've truly been infected but never got sick. That can happen. If everybody around you gets sick, but you got the vaccine of whatever disease and you don't get sick, you could probably say that's due to the vaccination, but it's not as easy. Drugs and vaccines all have unwanted side effects. Like even the best vaccines, there's going to be the potential for risk, but the risk must be weighed against the benefit. Because this vaccine side effects cause disease in healthy children, that's a tough pill to swallow. And so the standards are very high. They're set very high for vaccines. And they're always going to be higher than what we see for drugs, because drugs are given to somebody that's ill, and any alleviation is going to be a benefit. But getting a healthy child sick by a vaccine, that's not acceptable. Because of the nature of herd immunity, individuals can rely on others to get the vaccine and say, well, I'll let everybody else get the vaccine and then I'll just be protected by herd immunity. So you get all the benefits, but you don't share in the risk. However, it's kind of a selfish strategy. And if enough people take this selfish strategy, then herd immunity drops below a protective level. And then an epidemic could occur and lead to a lot of people getting sick. So I'm going to just end with this. Although vaccines are arguably the most successful of all medical advances to date, their acceptance has been greatly influenced by things that have nothing to do with medicine or science.